Cell cycle and cell division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that's formed by growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also take place. All these processes, that cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of a cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases: interphase, M phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of a cell cycle of a human cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of a cell cycle. The M phase starts with a nuclear division corresponding to the separation of daughter chromosomes karyokinesis and usually ends with division of cytoplasm cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, S phase, G2 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosomes at G1. Even after S phase, the number of chromosomes remains the same, that's 2n. In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult humans do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further, exit G1 phase to enter an inactive stage called quiescent stage of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending upon the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plant can show mitotic division in both haploid and diploid cells. M phase. This is the most dramatic period of the cell cycle involving a major reorganization of virtually all components of cells. Since the number of chromosomes in the parent and progeny cells is the same, it is also called equational division. Though for convenience, mitosis has been divided into four stages of nuclear division. Karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following four stages: prophase, metaphase, anaphase, telophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase. In the S and G2 phases, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The chromosome which had undergone duplication during S phase of interphase now begins to move towards opposite poles of the completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at centromere. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called esters. The two esters, together with spindle fibers, form a mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleoles, and the nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of centromeres are called kinetochores. These structures serve as the site of attachment of spindle fibers formed by spindle fiber to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from opposite pole. The plane of alignment of the chromosomes at the metaphase is referred to as the metaphase plate. The key features of metaphase are spindle fibers attached to kinetochores of the chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosomes move away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at leading edge, with the arms of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key events: centromeres split and chromatids separate. Chromatids move to opposite poles. Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows the following key events: chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope develops around the chromosome clusters at each pole, forming two other nuclei, nucleoles, Golgi complex, and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell and grows outwards to meet the existing lateral walls. The formation of new cell wall begins with the formation of a simple precursor called a cell plate that represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and blasters gets distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example: liquid endosperm in coconut. Mitosis, or the equational division, is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples why you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of gut and blood cells are being constantly replaced. Mitotic division is in the meristematic tissues. The apical and the lateral cambium results in a continuous growth of plants throughout their life. Meiosis. The production of offsprings by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. Four haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following phases: meiosis 1, meiosis 2, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following five phases based on chromosomal behavior: that's leptotene, zygotene, pachytene, diplotene, diakinesis. During leptotene stage, the chromosomes become gradually visible under the light microscope. The compaction of chromosomes continues throughout the leptotene. This is followed by the second stage of prophase 1 called zygotene. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure called synaptonemal complex. Synaptonemal complex. The complex formed by a pure synapsed homologous chromosomes is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived compared to the next stage, that is patch-teen. During this stage, the four chromatids of each bivalent chromosomes becomes distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of patch-teen, leaving the chromosomes linked at the sites of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the sites of crossovers. The X-shaped structure are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is diakinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to metaphase. Metaphase 1. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Telophase 1. The nuclear membrane and the nucleolus reappear. Cytokinesis follows, and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of interphase nucleus. The stage between the two meiotic divisions is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2, as much simpler prophase than prophase 1. Meiosis 2. Prophase 2 Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to Meiosis 1, Meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again becomes compact. Metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from opposite poles of the spindle gets attached to the kinetochores of sister chromatids. Anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosomes, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell by shortening of microtubules attached to kinetochores. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, results in the formation of tetrad of cells. That's 4 haploid cells. Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organism. Even though the process per se paradoxically results in the reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution.